In this video presentation, I will be going over the evidence of change, Pacific salmon, and how all of this relates back to our class. Evidence of change are fossils, embryology, homology, and vestigial structure. Fossils show how a species used to look before evolution changed it, and how it looks now. Embryology shows how a species has a common ancestor by similar embryos. An example of this is that many animals, including lizards and humans, used to have gills because they used to be fish, which means that embryos have gills. Homologies are similarities due to a common ancestor. An example of this is that a human arm and a bat wing have the same anatomy, but in a different shape. Vestigial structure are things that we don't really use anymore that our ancestors used to have, like our wisdom teeth. Pacific salmon. Pacific salmon have de-evolved from randomized fertilization. This makes it hard for the gene pool to be good. When salmon breed in nature, the strongest usually survive. Wild salmon are at risk of extinction because of food competition with the hatchery salmon. With less diversity, wild salmon are dwindling. Salmon from hatcheries have trouble reverting from the original traits. The extra salmon also greatly harm the ecosystem by pushing the carrying capacity of salmon. I think due to the overpopulation of salmon, the salmon population will be affected negatively. I think that hatchery salmon will drive out wild salmon, resulting in extinction of salmon. If humans can fix what we did to salmon, then the salmon could survive and possibly speciate. In our artificial project, we figured out that there were 4 H's that majorly affected how salmon run: hydro, harvest, hatcheries, and habitat. Hydro is affected by dams. Harvest is affected by how much fish are being eaten. Hatcheries are affected by how many fish the humans are producing. And habitat is affecting the actual lake or river itself. The artificial lab question that I chose to answer was, how do you think the dam affects indigenous fish populations and environment? I think that the dam takes important breeding grounds from the salmon and disrupts their life cycle, making it harder for the wild fish to be born, whilst the hatchery fish get mass bred. This creates an imbalance in fish diversity. But how does this relate to our class? In our Unit 4 targets, we learned about molecular clocks. This relates to salmon because the salmon are de-evolving due to fish hatcheries. Molecular clocks use DNA and genetic mutations to estimate how long ago a species was an ancestor. Predator Claw Lab. The Predator Claw Lab showed us how some species could be heavily affected by a mutation. The predators with forked claws got the most beans versus the chopstick claws who got the least. In the Skittle Bug Lab, we measured how a bug could survive based on the patterns of the bug's shell. The bugs were split randomly on their island into 2 separate sides. On one side, it was a desert, and on the other, it was grassy. The yellow bugs survived best on the desert, and the green bugs survived best on the grassy side. And how are these connected exactly? Unit 4 targets talk about how we can tell an animal is related to another. Fossils, embryology, homologies, and vestigial structure. Predator Claw Lab talks in depth about how different genes can help animals survive. Like how salmon have different genes to differently help them survive in different rivers and different oceans. The Skittle Bug Lab talks about how geological isolation affects a species. Similar to the Galapagos finches. They all came from the common finch and evolved into different species to be able to eat their island's seeds. They have big beaks to eat big seeds and small beaks to eat small seeds.